everyone to another episode of Nerd RX podcast, and I'm your host Barkha. And today we are going to talk about immunoprecipitation. And to walk us through what immunoprecipitation is, we have Erin Hughes. Welcome, Erin, to the show.
Thanks so much for having me. I was really excited for this opportunity.
Thank you so much for being here with us. Before we go into the topic, why don't you introduce yourself to our listeners?
Sure. So, as she mentioned, my name's Erin, and I have a bachelor's in science in biochemistry and molecular biology from Otterbein University in Central Ohio. Very, very small campus, like 2,000 people, so don't be surprised if you haven't heard of it. I immediately went into a PhD program under the Molecular and Cellular Biosciences umbrella program at Wake Forest University in North Carolina. And that's where I'm currently a fourth year PhD candidate in the biochemistry and molecular biology program. And I have the pleasure of getting to study a cannabinoid receptor interacting protein, or CRIP1A, and its interactions between the G alpha subunit of the heterotrimers and trying to figure out exactly what's going on with cell signaling.
Okay, wow, that's great. So, Erin, would you mind telling us why did you pick immunoprecipitation and what got you interested in it?
So, immunoprecipitation is one of those tools that can be used pretty much for everyone. But specifically with what I'm looking at, it's really helpful because it's going to let us look at protein protein interactions or protein interactions in general, whether it's between two proteins or between protein, DNA, RNA, etc. So, for my project, when we're trying to figure out how this protein CRIP is interacting with other proteins, how is it going to be, modulating cannabinoid receptor activity and all of that, it's been a really great tool, just because it's going to let us look between two different proteins. And we have immunoprecipitation versus co-immunoprecipitation, which is when you're looking for actual complex, which has also been very helpful.
Right. So, why is immunoprecipitation important and what is it actually used for?
So it's really important because it's going to let you look for known or even just suspected protein interactions in vivo and in vitro. So you can use cellular lysates from whatever cell line you want to use, whether it's a cell line that has endogenous levels of proteins or if it's a cell line that you've transfected in your protein. And because there are so many proteins within the body, within the cell, and they're all doing very, very different things, looking to see how these are interacting is going to let us downstream kind of modulate those interactions and even, for example, use it as a druggable target. So eventually, looking at how does this interaction occur and can we stop that protein protein interaction from happening, and maybe we can help with pain control or any sort of other health effect that you might want to go for.
Wow, so it sounds like you can use this technique in a lot of different projects. I mean, different types of experiments.
It's super flexible. You can it's use it for a lot of different things, a lot of different areas, pretty much as long as you have an antibody to a protein of interest and some beads, you're good to go.
Yeah, antibody, I think, is one of the issue with a lot of things because sometimes you don't have a good antibody and you're stuck there.
That's actually been one of my major problems recently when I've been trying to do my metaprecipitations for my project. Because you have to make sure that that antibody that you have is actually going to be to an epitope that's going to let you bind to your protein of interest. So if you have a protein that is, for example, binding to another protein or a complex on the N terminus, and then you have an antibody to the N terminus, that's probably not going to be able to be used as well because you're going to be blocking your binding site of your complex. So that makes it a little bit more difficult sometimes. And trying to get an antibody for a commercially available antibody for a protein is sometimes also very challenging. I'm currently waiting on a custom antibody coming from Nepal.
I am too waiting for an antibody. I've been like, it's not immunoprecipitation, but I'm working on my Western blots, and the CB1 and CB2 receptors are like making me cry. I'm like, I still have a good data for CB2, but CB1 is, I'm trying so many antibodies from different places, but none of them have actually worked for me. So,
I can tell you the CB1 antibody we use is from Everest Biotech.
oh, I'm gonna talk more about that after this episode.
Yeah, yeah, yeah.
Uh, yeah, CP1 has been a problem for me. So hopefully this works. Thank you so much for the tip.
Hmm.
Um, so I think in terms of Western blot, I my issues are first obviously finding the right antibody, and then the figuring out the dilution. So do you have the same issues?
So that is something that it's kind of thought about, but mainly for immunoprecipitations, you add typically 1 microgram of your antibody to you know, protocol I have from previous graduate students is between 100 to 500 micrograms of your protein. So in my case, that's going to be total protein within the cell because we're trying to look at this like endogenously within the system of the cell.
Okay.
So usually, I mean, that's just a typical. I have tried for lately. I've been having problems getting the immunoprecipitations to work 100% of the time. So I've tried changing around the amount of antibody used, but typically 1 to 5 micrograms of antibody.
Okay. Okay. That is good. That is figured out. So let's talk about the steps itself. So why don't you give us an overview for the entire process of immunization? Just an overview, like how do you begin, and what do you expect, and what are the various steps involved?
Sure. So typically, you're going to start with a protein lysate or homogenate or something, basically something from cells or even purified recombinant and a protein that has your protein of interest. And if you're looking for a complex, then well, you want to make sure all your other proteins are also in there. Um, so once you have that, you're going to want to make sure to do some sort of Bradford or a BCA, just something to get a rough estimate of how much protein you have in your solution.
Right.
Dilute it down as needed, or just make sure you don't have way too much protein compared to your antibody. So once you have that, you're going to add your antibody, typically 1 microgram, which oftentimes is only like a microliter or two of your, especially with commercially available antibodies based off of concentration. And so this is where the protocols can kind of differ based off of experiences, but in my experience, we have figured out that if you leave the protein plus antibody mixture in the refrigerator overnight rocking, you tend to get better binding.
Okay.
So I leave that solution in the fridge overnight, so at least 16 hours. After six, it doesn't seem to make a difference if it goes a little bit longer than 16 hours, as
Okay.
long as it's in there long enough for things to kind of reach equilibrium and give that antibody a chance to basically go sort around and find exactly what protein you're going to be looking for. Then the next day, we use agarose beads, which are protein AG plus beads, and those protein AG beads are going to bind to the antibody. I usually let that go for a couple of hours at room temperature, and then that's when you can pull, basically spin down in a centrifuge your beads, which is going to have your beads attached to the antibody attached to your proteins. Um, from there, you can do Western blot analysis and then stain for your proteins of interest. You can also use magnetic beads, and I know a lot of people have had good success with that. That's actually my next change to my protocol to try and get things 100% working is to swap over to magnetic beads, which are supposed to be
Wow.
really helpful too.
So are these beads like secondary antibodies?
Uh, eh, they kind of are. The protein AG is going to bind to the IgG and IgA of the antibody itself, so the long and short chains. Um, so in some ways, it is like a secondary, just attached to an agarose or magnetic bead.
Oh, magnetic bead. This is the first I've ever heard of. Could you tell us a little more about that?
Yeah, so they are, instead of agarose, which is what I typically use because it's cheaper, they're basically little tiny magnetic beads, balls, if you will, that have the protein AG attached to them, same as
Hmm.
your agarose. And then you can use a super magnet to basically pull the beads off to the side so that you can basically pipette or elute off anything that you don't want in your final solution because you're just looking for what stuck to your beads. So usually you're
Hmm.
trying to get rid of the stuff that didn't, which is going to be your supernatant in that case. So it's a really great way to, because the beads are stuck, so closely to the side of your tube, to make sure that you're not, pulling your beads out of solution too early, or mixing your bound, what's stuck to the bead versus unbound or supernatant. They are more expensive, which is one thing that some people don't typically use them for, but my
Hmm.
experience has been they're pretty awesome.
Oh, wow. So after you're done with the immunoprecipitation and you have your bead, you go for a second technique, like you mentioned, Western blot.
Yeah.
So does that mean, so when I do Western blot, I usually have my brain homogenates and I do a BCA to quantify how much protein we have, and then I just run my Western blot. So with this immunoprecipitation, what I am getting an idea is that you are kind of purifying it. You are just extracting your protein of interest, right?
Yeah, that's right. So it's like you're just trying to get that one specific protein out of out of a solution that has thousands of proteins. And in my
Mm-hmm.
case, I'm technically doing a co-IP where I'm looking for a protein complex because we're
Mm-hmm.
trying to see what Crip's interacting with specifically in our cells. So it's just a good way to kind of pull out your only protein plus whatever's hanging on to it versus Western, where you have way more proteins.
So your Western blots would look super clean.
Yeah, typically they are. You get a very strong band of interest for your proteins. Low background. Yeah, just it increases the signal actually quite a bit.
Right, yeah, I would imagine. Wow, now I'm considering leaning to this a bit more because the bands we get are super faint and we have a lot of background signals. So I think this is something that would help us. So, so just out of curiosity, for example, if I am trying to isolate CB1 protein, right? And I use an antibody in the immunoprecipitation and then, for example, a magnetic bead, and I isolate and put it on the Western blot. So, do you use this same primary antibody for immunoprecipitation and the Western blot?
So I have in the past, but that depending on your type of uh, it depends on if your antibody is covalently linked to your bead or not, because
Okay.
usually when you're trying to get your protein to basically pop off of your bead, you are going to heat it or going to use low pH or just something. And my experience has been our antibody then comes off too, off the
Okay.
bead as well. And it ends up being run on the Western. And some people it doesn't bother because the heavy light chain can run at a molecular weight that's not going to interfere with data analysis.
Mm-hmm.
But in my case, what I'm looking for actually runs at the same molecular weight as the antibody. So typically, my primary antibody, I try to use a different species, which makes it kind of interesting sometimes when you're looking for multiple proteins. We recently have tried using a chicken antibody, which actually works remarkably well.
Wow. Yeah, I haven't tried chicken. The only antibodies I'm working with right now is mice and rabbit. And I don't know, like I'm trying to find out CB1. They only have rabbit CB1 everywhere.
Yeah, luckily, we have a company that has goat CB1, which has been fantastic because that
Really?
gives another species for us not to have to worry about cross contamination with.
Yeah. Oh, wow. That would be super nice for me as well. Well, thank you so much for that description. And I can see how you can utilize this technique in so many different ways. So, what is co-immunoprecipitation? Because when I was actually reading up something on immunoprecipitation before our episode, I came across something co-immunoprecipitation.
So, co-immunoprecipitation is when you're looking for a protein complex. So, not
Okay.
just two proteins, but a whole bunch of them, if you will. So, I can use my research as an example with CRIP. We're looking to see if it interacts with G alpha I's, so the G alpha subunit of the heterotrimer. Now, when we immunoprecipitate looking for CRIP and beta, it suggested that CRIP's also interacting with G beta. So we have to kind of consider if co-IPing CRIP is going to get us, both G alpha and beta, as well as potentially gamma, and even looking to see if CB1 is in there. So co-IP is going to be looking for your actual complex of proteins, because we know a lot of times in cell signaling and in general cell processes that proteins are not doing their whatever they're supposed to be doing on their own. It's often happening with some other protein in some other part of the cell.
Okay. And has it ever happened to you that you're trying to uh, do immunoprecipitation for one protein, but you end up getting something else?
Yes, it has.
Okay.
And that oftentimes causes a little bit of a moment, because you're sitting there looking at data that doesn't quite make sense. And I
Mhm.
think, especially if you're Western blotting and then think of using a negative control and you're going to blot for a protein that you don't think should be in the immunoprecipitation but does come up, has happened, and it kind of has led us to want to send our samples off for mass spec just to see how much is actually coming down in our immunoprecipitations with grip.
Okay. Do you have any alternative techniques that you can use instead of immunoprecipitation?
Yeah, so since you're looking for a protein protein interactions or things like that, you can run BRET or FRET, which is going to be looking for fluorescent signal when two proteins get close together. I am It's just things I have heard that I should be doing for my committee, so I have not really looked too much into those yet and whether or not we're actually going to use them or have much information on them. What I do in parallel as well is I do cell imaging. So I plate my cells on cover slips and then stain for CRIP plus whatever antibody, what other proteins I want to be looking for, and look to see if there's co-localization under the microscope. And I use a few other techniques as well. Typically, you can, I would say, running in parallel your Brett Fret cell imaging. You could even take your immunoprecipitation and put it on, do mass spec with it after instead of Western blotting, just to get your exact molecular weight of whatever proteins are floating around in solution.
And is this technique user friendly? Like, how long will it take for a completely new person to learn this?
So I think it's pretty user friendly because there's a lot of protocols out there, and even a lot of companies have their version of the protocol available. So there's typically a little bit of troubleshooting, but I would say it only took me probably a couple weeks to get really, really familiar with the technique. Now, obviously, there's always going to be troubleshooting with anything that you're doing in science.
Always.
Um, Troubleshooting's what takes a while, but I think it's pretty simple because it's you're mixing beads plus a solution and doing a lot of centrifugations. The centrifugations get on your nerve after a while.
Yes, yes, yeah. I think troubleshooting is the main part of all the experiments. And I think you spend so much more time on troubleshooting than your actual results. You just have to get right once. So, troubleshooting is a big thing everywhere, I feel. So let's talk about the advantages and disadvantages of this technique.
Yeah, the advantages would be that it's very flexible, especially if you're using a just an agarose bead with protein AG, or a magnetic one with protein AG that's going to be able to bind to any sort of antibody that you have in solution as long as it has an IgG or an IgA on it. So that means that if you're looking at a wide variety of different proteins, or your project is very diverse, you can kind of mix and match your proteins of interest with whatever antibodies you want to use. Now, the disadvantages being, like I said, sometimes your antibody might not bind the way that it needs to be, or your immunoprecipitation isn't working quite right, or you're just not seeing signal on your downstream, imaging, your quantifications. So there's this kind of pros and cons to it, just like everything. But I think my main attraction to it was being that I could pretty much look for whatever protein with whatever antibody I want to, and it's great for working parallel back and forth. So if you're immunoprecipitating for CB1, for example, and you find that it interacts with the G alphas, then you can also immunoprecipitate with the G alphas to see if you get CB1. So you can kind of work in reverse there.
Okay. Wow. So, have there been cases where immunoprecipitation cannot work?
I have actually found that, at least with my methods, that certain detergents can block some interactions of proteins with the antibody, which is not ideal when you because a lot of cell lysis buffers actually have detergent in them. That's how you burst open cells.
Hmm.
So in that case, I've had a time or two where the IPs just didn't work. There was no signal, and I couldn't figure it out for a while. Um, or your antibody doesn't bind to your protein like it's supposed to. That's the other case where it definitely wouldn't work.
Okay, and this can be used even with tissue homogenate, right? And not just cell lysates.
Yeah, I would imagine so. I have not tried that. I think it's something that my advisor and I have talked about briefly. We currently don't have any animals or any brains for us to work with, but I definitely would say that you'd be able to do it. I think as long as your proteins are, available for the antibody to find and bind. I don't see why it wouldn't.
Okay, awesome. And what would be the cost associated with it? Like, is this a technique that any lab can set up easily, or is it expensive?
I would definitely say it's very low cost. Especially if you're a lab that already has cells or brains or tissue homogenate already up and running on your day-to-day basis. The especially the agarose beads, instead the magnetic ones can be kind of expensive, but if you're trying this initially just to see if it's a technique you want to use, the agarose beads are very, very low cost. Well,
Okay.
science is expensive to begin with, but they're low cost in the world of science. um, Especially, I think Santa Cruz Biotech has protein AG beads for like $90
Wow.
for a milliliter of them, and you don't use that many. So,
So that is another advantage, I would say. It's easy to set up. So I think my last question for you would be, is there any interesting articles or protocols out there that I can link down in my description for our listeners to read up on?
yeah, so there's when I was really digging into co-immunoprecipitation and immunoprecipitation, I came across this methods in molecular biology paper from 2017 that's just very simply titled protein-protein interactions co-immunoprecipitation. And I thought it was a great read just because it talked about how you can use genetic, like yeast to yeast hybrid, and biochemical, so your co-IP methods, to basically look to see what proteins are interacting together. And it gave a really good protocol and background. That was, I think, one of the first papers I ended up reading about.
So, well, with this, I think we are going to end today's episode. Thank you, Erin, so much for being here with us and explaining what immunoprecipitation is.
Yeah, thanks so much for having me. It was great talking with you.
Likewise. And listeners, I will catch you next week for another episode on this podcast. And in meanwhile, if you wish to suggest, if you have any suggestions for any technique, or if you would like to join me on podcast and discuss a technique, please email me at barkha@nerdrxpodcast.com. And remember, it's good to be a nerd. Bye.